everyone, and welcome to today's webinar. This webinar is hosted by Jen and sponsored by 908 Devices. Our topic today is rapid clone screening of biosimilar candidates using microfluidic CEMS. Thanks so much for being here. I'm Utrecht Thomas, Senior Editor with Jen, and it is my pleasure to host today's webinar. We have two great speakers lined up for you today. We have Dr. Erin Redman, a principal scientist at 908 Devices, and Ruben Cageling, a doctoral candidate at Polpharma Biologics Utrecht and VU Amsterdam. We'll get to their presentations in just a moment, but first of all, I'd like to cover some background on today's topic. Developing a biosimilar drug requires screening samples originating from different cell lines in order to select the most appropriate ones for further process development. Each sample needs to be characterized by their critical quality attributes, such as glycosylation or charge variants, and these can be used to identify potentially harmful modifications in the samples. Some common techniques used to determine the charge variants of a sample, such as ion exchange chromatography, are time and material consuming, and they typically use optical detection to determine the relative peak area of acidic and basic variants. Now, an alternative approach for assessing charge heterogeneity is microfluidic capillary electrophoresis coupled to mass spectrometry, or CEMS. The microfluidic CE technology boasts low sample consumption and short analysis times, while the hyphenation to MS enables identification of the charge variants and the glycosylation profile of the native protein. In today's webinar, our speakers will describe how the ZipChip microfluidic CEMS technology enables rapid early-stage clone screening for biotherapeutics development. They will present a case study where the ZipChip was used to select clonal cell lines based on their charge variants and glycosylation profile. Furthermore, they'll also highlight the new CVA kit, which enabled better separation and sensitivity in charge variant analysis of biotherapeutics. So what are the key takeaways from this webinar? Well, you hear about microfluidic CEMS technology basics and applications. You'll hear about the benefits of combining charge variant separations with mass spec characterization. And you'll hear about how this technology is an asset for rapid clone screening of biosimilar candidates in the biopharmaceutical industry. Today's presentations will be followed by a live Q&A session. If you have a question for our speaker, you can enter it at any time in the Ask a Question panel located on your screen. All right, let's get started.
Hi, thank you for that introduction. So in my portion of the webinar, I'm going to start off with covering a bit about the ZipChip technology. I'll introduce our charge variant analysis assay, cover a few key applications in biopharma, and then I'll pass it on to Ruben to talk about how he uses ZipChip in our charge variant assay for clone screening and biosimilar development. The ZipChip platform is a simple CE interface for commercial mass spectrometers. It consists of the interface, the chips that perform the separation, and assay kits. The interface fits onto the front of mass spectrometers just like a source, and it makes all of the necessary gas and electrical connections when it's installed. The chips perform capillary zone electrophoresis separations, and the separations are directly integrated with electrospray ionization from the corner of the device. There's a few different varieties of chips, and they're all optimized for specific separations. Our assay kits include background electrolyte that you need for the analysis and sample diluent for sample preparation. These are optimized for specific applications, and it reduces the amount of method development needed to get started. Like I mentioned, with ZipChip, we perform our capillary zone electrophoresis separations using microfluidic technology. This is very different from your typical capillary-based CE system. Our chips fit into the interface where it makes all of the fluid and electrical connections. And in the middle here, I have a diagram that shows the schematic of the chip. By using microfluidic technology that allows us to integrate multiple different functional elements into our device design, and that really speeds up analysis, allows us to generate robust electrospray and perform high-resolution separations seamlessly. After a sample is injected here at our injection cross, it enters the separation channel, and we apply voltage, and that creates a field strength for the separation and also initiates electrospray at the corner of the device. The sample analytes migrate through this serpentine separation channel. This channel is analogous to the pulled capillaries that you may be familiar with from traditional CE systems. As the analytes migrate out of the separation channel, they're ionized in the electrospray plume and enter the mass spec. Again, we're performing capillary zone electrophoresis separations. So our separations are occurring free in solution. We're not using any stationary phases or polymer matrices. This type of separation is well suited to a wide variety of samples, from small molecules all the way up to large intact proteins. And in the life sciences industry, ZipChip's being used for quite a number of different applications. But in this presentation today, I'm going to focus on intact protein separations, and specifically analysis of monoclonal antibodies. For charge variant analysis with ZipChip, we recommend using our charge variant analysis kit paired with an HRN chip. The CVA kit is a fairly recently released product, and it offers improved sensitivity and ease of use compared to our previous kits for antibody analysis. And in Ruben's presentation, you'll see data from this kit and also some from our legacy kits. With these analysis conditions, you can generate charge variant separations that are comparable to other techniques like isoelectric focusing or charge exchange chromatography. On the left here, we have a separation of the NIST-MAB reference material. And you can see the antibody is separated into two basic variants, the main variant and acidic variant. But a major benefit of the ZipChip CVA application is that it's integrated with mass spec detection. So for every peak in the charge variant profile, you can obtain a mass spectrum. The molecular weight information you get from the mass spectra is then used to identify post-translational modifications as well as glycoforms. And you also have the charge variant profile to look back to make sure those post-translational modification IDs actually make sense with what you're seeing in terms of charge heterogeneity. Here's an example where the mass spec data is really impactful. Based on just the charge variant profiles of these two antibodies, they look to be very comparable. You have the same number of basic variants, the same number of acidic peaks, and they're in pretty much the same distribution. With just optical detection, that's all the information you may get, and you may stop there and conclude that they're more or less the same. But when the mass information is considered, it's obvious that these two MABs are actually quite different. On the left here, we have the NIST-MAB reference material, and the basic variants are from C-terminal lysine clipping, and we can tell that there's nearly 100% conversion of the N-terminal glutamine to pyroglutamic acid based on the mass that we're measuring. And the acidic species for this molecule is primarily due to deamidation, as we observe a pretty minimal mass shift for that peak. However, this NIST-MAB that's expressed from the NIST cell line has basic variants that are different. The basic variants are from N-terminal pyroglutamic acid formation rather than C-terminal lysine clipping. And in this case, we actually see that we have nearly 100% clipping of the C-terminal lysines. We also see some differences in the acidic peak. It has a much larger mass shift, so this is likely a combination of multiple modifications like deamidation or glycation. When we look at the glycoform profile, we can see there are even more differences. For the NIST-MAB reference material, it has more extensive galactosylation on the glycans than we see with the NIST-MAB expressed from NIST-CHO. And with the NIST-CHO expressed MAB, there's much more signal from incomplete fucosylation of the glycans. So with just one simple assay, you're able to see quite a number of different product quality attributes for these molecules without doing extensive characterization with other techniques. To date, our customers have mostly implemented our charge variant assay to support downstream processing and product characterization in the biopharmaceutical industry. But recently, we've been exploring how to implement ZipChip charge variant analysis in a more upstream way to support bioprocessing. We're able to use the ZipChip in a dilute and shoot manner for analyzing antibodies directly from cell culture. This allows us to monitor product quality attributes in a simple, easy way earlier in the development process. On the right is a charge variant separation of NISMAB expressed from the NISCHO cell line, and below it is the process mass spectra for the main variant. For this analysis, we took media directly from the bioreactor and simply diluted it 10x with the sample diluent from the charge variant analysis kit. During the separation, media components that may interfere with analysis are separated away from the MAB, so we get a nice separation as well as high-quality mass spectra. In this case, we're able to separate two basic variants, and again, an acidic variant, and identify the basic and acidic variants from the mass data, and also assess the different glycoforms on the antibody. If we analyze samples from throughout a bioreactor culture, we can actually observe changes to the antibody product as it's expressed. On the left, we have a figure showing the charge variant distribution at various points throughout a 14-day bioreactor run. And on the right, we have a similar figure showing the glycoform distribution. Early into the culture, we can see that there's a higher percentage of basic variants than at the end. We can also confirm from the mass data that the identity of the basic variants and the acidic variants remain the same throughout the culture. For the glycoforms, early in the culture, there's a higher percentage of incomplete fucosylation on the glycans, but by the end of the culture, we see that as reduced, and we have a higher percentage or more extensive galactosylation of the glycans. Monitoring the antibody product in this manner provides the potential for further process refinement of the bioreactor run, and also gives you the chance to intervene earlier if a batch is not meeting key quality requirements early in the growth process. So I hope I've shown you that ZipChip is a simple way to perform high-quality CEMS charge variant analysis. With the separation and mass detection, you get multiple pieces of information to characterize your antibody and monitor product quality attributes. This type of analysis can be an asset in biopharma because it allows for both in-depth product characterization downstream, as well as the potential to support upstream processing with direct from bioreactor charge variant analysis. And now I'll let Ruben discuss his application of ZipChip in clone selection and biosimilar development.
So hello everyone. My name is Ruben. I'm a PhD candidate at Polpharma Biologics in Utrecht and for the VU University in Amsterdam. And today I'm going to talk about rapid clone screening of biosimilar candidates with microfluidic CMS or the ZipChip. First, a little introduction. So first, what are biosimilars actually? A biosimilar is a biological drug which is highly similar to another already approved biological product for which the patent exclusivity rights have been expired. This drug is highly comparable to the reference product, but always taking into account natural variability because it's a biological product. And it also should have no clinical meaningful differences between the biosimilar and the reference product in terms of safety and quality and efficacy, meaning that both drugs can be used interchangeably. So what are the benefits of this? First, of course, you have the same quality as the reference product. This means also that you have to do less clinical studies. You only have to prove that they work the same, meaning that the product, you already have a target, so it's quicker on the market. making it therefore also cheaper compared to the reference. And this also opens markets worldwide, making it more wider available because of the lower price points. And some very famous examples are insulin and monoclonal antibodies. And this monoclonal antibodies is mainly where Polpharma is well versed in. So Polpharma Biologics, Polpharma itself was established in 1935 in Poland as a company that develops and manufactures small molecule medicines. And in 2013, the owner decided to start a separate company called Polpharma Biologics to develop and manufacture biosimilars. And as I said, the main focus here is on monoclonal antibodies. And currently, as you can see from our portfolio, we have 2 products on the market and many more to come. So we are located at several places. Our group holding is located in Amsterdam, while I am located in Utrecht. This is really the R&D center where we set up cell lines and start up the early process development. We have a joint venture for our ranibizumab antibody, which is located in Switzerland. And we have 2 big manufacturing sites in Gdansk and in Warsaw, where they also do a lot of process development. So how does this pathway go typically? Well, you always start with the cell line. We select the best cell line, and then you go on with further processes for upscaling and purification, transfer it to a different site, and there we do drug product formulation, etc., fill and finish, in the end, bring it to the patient. So as I said, I worked in Utrecht, and here we are mostly focused on the first 3 steps. And in the analytical department, of course, you need to support all these phases, because when you generate a protein or when you create a monoclonal antibody, it's very important to keep track of the quality of the protein. So here we do assay development and we do sample analysis for all steps in these early stages, like upstream and downstream processing. And we monitor the critical quality attributes of the products, like N or C terminal heterogeneity, side chain modifications, glycans, disulfide bonds, and more. And there we use liquid chromatography techniques like ion exchange or size exclusion, capillary electrophoresis techniques. And of course, the ZipChip is also in this category. We use mass spectrometry and other techniques like binding ELISAs or other type of bioassays. So where does the ZipChip come in for the cell line screen? Well, many cell lines are set up in early stage cell line development. And when you set up many cell lines, the more you set up, the bigger the chances in finding the best producing cell lines, which you can use for later stages. When you set up these cell lines, you all need to screen them based on their critical quality attributes. And these cell lines we also call clones, the samples we get from them. And you check them, for example, on titer, fragmentation, glycosylation, and charge varieties. And I want to focus a little bit on the last 2, because for glycosylation and charge varieties, we use multiple techniques. Most common techniques that are used are cation exchange chromatography with UV detection, or release-stained glycans for the glycosylation using helic separation with fluorescence and MS detection. Even though both techniques are an industry standard, they also have its pitfalls because they have long run times, they have preferably high concentrations that are needed. For the release glycan assay, you need a laborious and costly sample preparation. And for KEX-UV, you only quantify actually based on relative abundance of acidics and basic species. So you don't know what is behind there. So we come up with the question of what if we can determine the glycosylation profiles and the charge varieties in one analysis of these clones, and that we can also identify actually what are these charge varieties. So this is where in Polpharma we really employ the ZipChip. The ZipChip, as of course already discussed, is a capillary electrophoresis device, and it really helps us to do high throughput charge variant analysis on a microfluidic chip. That's also why I sometimes call this MCEMS. And the benefits are really for us that it uses almost no sample, only 1 nanoliter per run. And if you look in the left bar chart, you see that this amounts to only 0.5 nanogram per run, which is way lower than all the other techniques we use more in the micrograms of material. It also has short analysis times, 10 to 15 minutes per run. Again, that's at least 4-fold quicker than other techniques that we typically use. And of course, because it does not use a lot of sample, and yeah, this is also something that we really need in these early stages, because there's also not a lot of sample that we gain or that we get from the cell line development department. And an additional benefit, of course, is that we now, because it's coupled to the mass spectrometer, we can identify the charge variants, and we use this to determine the major glycoforms and modifications. So how did we do this? So this is a part of the stuff that we did for my first paper that we recently submitted, and hopefully is available soon. So the samples that we used was an in-house IgG1 biosimilar candidate, and we used 5 different monoclonal cell lines. We took samples from this, and we call them clones for now on, and we compare it to the originator molecule. The samples were then protein A purified, they were buffer exchanged, and diluted to 0.5 milligram per milliliter, and we did this in triplicate. And as setup, we used the ZipChip interface coupled to an autosampler and to an Explorers 240 Orbitrap MS system from Thermo. We used the high-resolution native or HRN chip, which has the longest channel length to ensure better separation. As BGE, used the native antibody BGE plus 4% of DMSO to boost the signal. The runtime of each sample, including the conditioning step, was 50 minutes in total. So that's very quick. And for data analysis, we use Chromeleon for the electrophoresis and use sliding window deconvolution in Biopharma Finder to assess the mass spectrums. So the goal was really fun. How can we use the ZipChip in clone screening? Well, let's first look at the profiles. So as you can see, we have 6 different profiles here. The basic species migrate before the main peak because they have a higher electrophoretic mobility, because they are more charged compared to the main peak. Or conversely, the acidic peaks migrate after the main peak because they are less charged compared to the main species. And what you see here is that the peaks are very well separated. You can clearly see the distinguishing. You can clearly distinguish between the basic, the main, and acidic peak. And from the triplicates, we could deduce that we have very reproducible migration times with low RSD values, and also the relative peak areas after integration are very low in RSD. So if you would do ion exchange with UV detection, you would stop here because you have your percentages acidic and basic. But now we really look into one of the benefits of this method, and that is actually determining what is behind each peak. What is each charge variant? So as basic species, we found 2. In clones 1, 2, and the originator, you found C-terminal lysine. And C-terminal lysine is a modification that is a post-translational modification. The C-terminal lysines are cleaved off mostly by carboxypeptidases in vivo, both in the body, but also sometimes during cell culture. So this modification doesn't really have a clinical relevance. The other basic modifications that we found in clone 3, 4, and 5 was C-terminal protein amidation. This happened also at the C-terminus, and when the C-terminal glycine is clipped enzymatically, the C-terminal protein gets amidated, and this also causes an additional charge, to the monoclonal antibody and making it a basic species. So if you would look at all the profiles, you would think, oh, they all have the same basic species, but actually now with with the use of DMS, we could determine that they are actually different. Even though both species don't have a clinical relevance, this is also true for the protein amidation, because it also happens a lot in endogenous IgGs. Still, we could use this information to get more depth and more knowledge about the products that were actually created. And so the acidic peak were also identified in all of the samples as asparagine deamidation. And here, the asparagine is deamidated into an aspartic acid, which then reduces the charge, and this causes an acidic peak to appear in the mass or in the charge profile. And if we then look at the glycosylation, because we can also determine the glycosylation profile, and we mainly use the main peak for this, we found a wide variety of glycoforms detected for each clones and for the originator. You can, for example, see that clone 1 and clone 2 show very distinct glycoforms compared to the other clones and the originator. What is also apparent is that there are notable differences between the glycosylation and afucosylation of the clones and originator. And the afucosylation, so the degree of glycoforms that are here that don't show any fucose or only have fucose at one of the two glycan sites, is here important for this antibody on the study, because this this relates to the mode of action of this particular biosimilar candidate. Also, the galactosylation is mostly a very important critical quality attribute, and you can see that all of the clones are not close compared to the originator. Well, now we have all the information that from the analysis, so now we can make an actual cell line selection. So what we did, we compare all the results of all the clones to the originator. We take the originator range and one standard deviation from the triplicate, and we see if the samples fall within the range or outside of the range. And then we give a score between 0 to 1 based on the closeness of the originator. In addition, samples can also be rejected directly if we would have found modifications that could be potentially harmful. So you can see in the graphs on the right that for each of the critical quality attributes, these values are plotted, and you can see that some are in the yellow or in the green range or in the red range. And these colors mean the following. So if it's in green, the sample is in range, and then it gets a score of 1. If it's in yellow, then the sample is outside of the range, but still this value could be acceptable. So we give it also 1. And this is the case with deamidation, for example, in acidic variants, because deamidation tends to increase over time during process development. So this value is even more desirable maybe than the values that are already in range. If a sample is in red, then the sample is outside of the range and you get a score lower than 1, and we calculate the score based on how far it is off of the originator range. And finally, we multiply these values with a weighing factor based on how critical these CQAs are. So as I said, afucosylation relates to the mode of action of this molecule, and therefore was weighed with a factor of 3, while the other ones are all weighed with a factor of 1. And this is then the end score that you would get. And from here, from the total sum of the score, you could see that clone 3 scores the highest and would so potentially be interesting for further process development, while clone 1 and 4 could also be very of interest because of their closeness also to clone 3. Clone 2 will definitely fall off the wagon in this case. Then we also performed a transferability study. So we wanted to see if this method is also transferable and can also be used somewhere else, and if you then would get the same results. So we transferred the samples to NIBRT, the National Institute of Bioprocessing Research and Training in Dublin, Ireland. We shipped the exact same samples, and they were tested in triplicate in their lab using the same sample preparation, same method, and same instrumental setup, so same mass spectrometer. And from this, as you can see in the figures, we obtained similar results between both labs. They don't differ that much, and we only saw a small difference in the percentage of charged ions, for example, the acidics, This can happen due to sample handling or sample storage. In the end, we also try to see if we do the scoring again, if we would pick the same clones, regardless of the laboratory that it was performed. And as you can see in figure D, this is the case. They almost score exactly the same based on regardless of the lab that it was performed. So we could say that the method was transferable. So to conclude this part of the study, so the ZipChip for us is really a valuable tool for rapid clone screening of early stage clones. And one of the benefits is that it can measure 96 samples a day. And as I said, these sample types numbers are typically high. So this is really valuable to get the data quickly. And with other techniques, you would end up with 22 to 24 samples a day. The ZipChip only requires a small sample volumes, and you can even retrieve it. And this is also very helpful for these early stage clones, which don't have a lot of volume to begin with. It helps us also to get additional information based on the identification of the charge variants, and we can use this information to guide decision making. For example, we used also the ZipChip to determine that we wouldn't continue with the project based on the identity of the charge variant that we found with the ZipChip analysis. And finally, the method proved to be transferable between two sites, and we can really use this information to enable better process development and guide it better, because we get a lot more information in less time. Well, then I would like to talk a little bit about something else. Thanks to 908 devices, we're also able to test the charge variant analysis kit. And this we did in 3 different experiments. So the first experiment is a dilution series in which we took the NISMAP sample and we diluted it to concentrations between 0.0008 milligram per milliliter to 0.5 milligrams per milliliter, and tested it using the standard native antibodies with the MSO BGE, the CVA kit BGE with and without the additive. And then we expanded this a little bit more. So we also checked this for 1 concentration for 2 in-house samples of us, because we wanted to see if it not only works for NISMAP, but for other samples. So we took 1 IgG1 and 1 IgG4 sample. And again, we checked with the native antibodies BGE, and in this case, only the CVA kit without additive. Finally, we performed a small clone screening study, again, a sort of case study as you've seen before. Here we took 2 samples from clonal cell lines, and this time they were producing IgG4 biosimilar, and we also used the originator molecule to compare it. Samples were protein A purified, buffer exchanged into the CVA kit diluent, and then diluted to 0.05 milligram per milliliter. Again, a typical concentration that you would have during clone screening. As BGE, we used the CVA kit with the additive. And the setup was exactly the same as the previous study. So yeah, let's first show the results. So here are the electropherograms of the dilution series, and you can see that actually for all 3 BGEs, you would get even at the lowest concentration, would get signal, and you could easily distinguish between acidic, basic, and main peak. However, what is clear is that from the CVA kit, you could better distinguish the second basic peak in the electropherograms, which is hardly visible in with the native kit, and the overall peak shapes are better. For example, you can notice that the acidic peak is better separated in the CVA kit, and overall the peak shapes look better. Fortunately, there was some problem with the CVA kit plus additive at 0.5 milligram per milliliter. But other than that, the results for all conditions look very good. And this really helps to measure these low concentrated samples that we typically encounter. If you then look to the mass spectrum, so in this case, I took the mass spectrum of the main peak for both the CVA kit with and without additive to illustrate what the difference is between using the additive. And in this case, you can see that the additive allows for a more native charge state distribution. And there are also more charge states available. For example, you can see that it goes up to 19 plus, and the most intense charge states shift from 30 plus of the CVA kit to 27 plus when you use the additive. When looking at more at the individual variants, for example, I took the 29 plus charge state, you can see that there's no difference in the amount of species that you would detect. So the additive really helps you if you have a different kind of mass spectrometer, and you can use a more native range in the charge state distribution. So then we compared the native antibodies kit with the CVA kit and without the additive in this case. And you see for both NISMAP, the IgG1 and the IgG4, we got good results with both kits. However, the peaks are better resolved, especially the lower abundant ones. For example, in the NISMAP sample, you would see that small peaks are visible as the second basic peak. And in the IgG1 and IgG4, you see that the lower abundant peaks are better separated, and they are better distinguishable from the baseline in this case. And then the final part of this experiment is the clone screening. So as I said, we took 2 clones and originator sample, and we successfully measured them using the new CVA kit at a concentration of only 0.05 milligram per milliliter. And on the right, you can see the profiles, and you can see the differences between the originator and the clones, for example, in the basic species, which are way lower in the clones compared to the originator. If you also look at the percentages of area, you can see that, for example, in the acidic, these are much higher in the samples compared to the originator. So purely based on these percentages, you can see that they are not in range with the originator yet. But we also want to know what the identity is of these samples, of what is behind the charged variants. So in this case, there were more acidic species. We first found in all samples and the originator also the sialylated species. So this particular glyco form adds or reduces a charge in total, and then migrates as acidic peak. And you can see that the glycoform G0F, G1F with this NOAC sialic acid is higher in clone 1, while only in clone 2 and the originator don't have a lot of this of these sialylated species. For clone 2, we also found an advanced glycated end product, also called an H. In this case, it was carboxymethyllysine, which adds 58 Dalton. And this product is really comes from glycation. So glycation is the non-enzymatic addition of a sugar to the backbone of the protein, and then due to oxidation or other processes, this is chemically degraded into these advanced glycated end products. So we found it at very low levels, and depending on the location, you could it could maybe influence the binding a little bit. However, this information is currently not available. And of course, we also found deamidation, but because of the very small mass shift of only 0.98 Dalton, it was very hard to accurately quantify it. And if you really want to locate, of course, where it is, it's better to do a peptide mapping. If you then look at the basic species, we found 2 basic species. So we have the C-terminal leucine amidation after glycine clipping. This is the same as the proline amidation after lysine clipping. However, because this is an IgG4, the proline in the sequence is replaced by leucine, and therefore the leucine gets amidated. But the mode of action on how this happens is the same. And the N-terminal pyroglutamate, so the main peak has the N-terminal pyroglutamate at both heavy chain ends, but when this is not completely formed, so that you still have the glutamate without that is not cyclized, then you will also get an additional charge. And again, this resolves as a basic species. So here you really see the differences that we also already saw from the profiles. For example, the N-terminal pyroglutamate that is only happening at 1 heavy chain it goes up to 36.9% in originator compared to only 5% roughly in clone 1 and so almost barely there in clone 2. So here the difference are very visible, even though probably this won't affect the binding as much of the target. However, this also always has to be checked by doing, for example, a binding study with an ELISA. And we can also look at the glycosylation profile, of course. And in this case, you could see that clone 1 is different compared to clone 2 and the originator. Clone 2 and the originator really match already in G0F, G0F, and in G0F, G1F content. Well, you can see at clone 1, this is still off. Therefore, you would need to modulate, so you need to do more process development to bring clone 1 into the level of the originator. So in the end, from these, based on glycosylation, you would select clone 2. So in this study, of course, we cannot really put a lot of discussions because we only took 2 clones, but this again highlights that we can do clone screening at low concentrations with the ZipChip, and only based on 3 runs, we already get a lot of information in which we can determine what modifications are the ones that we want and what that we want to match, and which ones can we use in later stages, which ones are the ones that we don't have to adjust in the future more compared to the other ones. So, and this was all measured with the CVA kit, and the CVA kit is really easy in use. For example, if you don't use the additive, you don't need to add DMSO or anything. You can just put it on your system and go. And if you want to adjust your mass spectrum, your charge state distribution, it's very easy to just add a little bit of the additive. You don't need to buy a separate brand of bottle to do this. You just use the bottle that is supplied with the kit, and it's very easy to do. And what we also noticed from all the profiles that we saw is that the new CVA kit enhances the sensitivity of the ZipChip, and you could go to concentrations as low as 0.8 microgram per milliliter and still get a very usable profile, which is of course extremely low concentrated samples. You get very well separated electrophoreograms, and like I said, the samples that we measure at low concentration, we could get a lot of information out of this. So overall, we are very happy with the CVA kit, and we're now using it as a standard in all our current workflows. And I want to give a special thanks to my colleagues from Polpharma, Karin and Anja, to the persons at NIBRD that really helped us with the transferability study, which are Sarah and Jonathan, my promoters at the VU, Govert and Kevin, and of course the people from 908 Devices. So big thanks to Erin, Scott, Will, and Simon. So this concludes our presentation, and we are happy to take any questions that you may have.
I'd like to thank our speakers for their informative and interesting presentations. We're going to begin the live Q&A session in just a few moments, so please submit your questions if you haven't done so already. Before we start the Q&A, a reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please keep an eye on your email, and please do share the link with your colleagues who might not have been able to tune in today. All right, give us a couple of seconds to transition. And welcome to the Q&A portion of today's webinar. Thank you all so much for joining us. We hope you enjoyed those presentations. Our speakers are ready to take your questions. So I would just like to remind you that if you do have a question, you can enter the question at any time in the Ask a Question panel on your screen. We're going to be on here for the next several minutes taking questions. And thank you, Erin and Ruben, once again for those presentations. I'm going to jump right in and start getting through some of these questions that we have here. First question I have, and Erin, if you could take this one. The MS is not part of the 908 instrument, is it?
Right. For ZipChip, the hardware fits on existing mass specs that you would have in your lab. We do have a product called Rebel that uses the chip separations and integrates a simplified mass spec for amino acid analysis on the biomanufacturing floor. So if you're thinking of an integrated separation and mass spec box, that might be what you're thinking of.
Excellent, thank you. And Ruben, I think this one might be yours. Are you using the auto sampler? As far as I know, the minimum sample volume is 10 microliter compared to 1 nanoliter.
Yes, that's correct. I'm using the auto sampler, and indeed you are right, you need at least 10 microliter of sample volume. However, I was referring to the injection volume. So compared to the other techniques that I showed, the ZipChip only uses 1 nanoliter of volume, which I think is very low compared to the other ones. But indeed, you need at least 10 microliter of sample volume.
Excellent, thank you. Next question I have, does the glycosylation data obtained with the ZipChip correlate with other methods, such as the released end glycan assay? And Ruben, if you could take this one.
Yeah, sure. Yeah, indeed. We checked in our paper that is there a correlation between the data that we get with the typical and released end glycan assay, and we see that all of the major glycoforms that you will find with the ZipChip are also there with the released glycan assay. And even though the released glycan assay shows to be a little bit more sensitive, especially when showing low abundant glycoforms, the trends and the percentages of the glycoforms were the same across both the ZipChip results and the release and glycan assay.
Excellent, thank you. Do different medias impact the ability to do dilute and shoot measurements from cell culture? Erin, could you take this one?
Sure. So we're pretty new to doing dilute and shoot still, and it's possible that some medias may require more significant dilution than the 10x that I showed here. But so far, 10x dilution has been working pretty well for us for a couple different types of media that we've used.
Excellent, excellent. Next question I have, for all of the data shown, it appears that a biopharma version of the MS is used. Is this a requirement for running this assay? Erin, I'll come to you.
Sure. No, it's not required to have a biopharma enabled mass spec, though those do give the nicest results when you're talking about Orbitraps. This assay can also be done on time of flight instruments, which their ion optics are a little different, so you don't have that biopharma versus non-biopharma designation. And the assay should give good results across a variety of different mass specs.
Excellent, excellent. And just to remind our audience, we're taking questions live. So if you do have a question, please go ahead and enter it at any time in the Ask a Question panel on your screen. Next question I have here, when may it be necessary to do sample cleanup versus just diluting the media? Erin, I'll come to you for this one.
So if the titer is very low of the media that you've collected, perhaps it's very early in the cell culture process, it may be necessary to do a buffer exchange instead of a dilution just to make sure you have enough of the drug product material in the sample to look at. It's also possible that if the media is perhaps particularly salty or contains an unusual compound, that it may be necessary to clean it up then, but I think that's less likely.
Excellent. Next question I have, how early in the clone screening process do you think this assay could be used? Ruben, can I come to you for this one?
Yeah, definitely. Yeah, I think you can start very early on already with using the ZipChip. At our site, we already try to start early on with these measurements. So like in the really early phases that you just set up multiple clones, then it's especially helpful because then you have a large number of clones. You want to measure them, but with the standard techniques, you're either not able to, or it takes a very long time. You have long run times, 45 minutes, like I showed you. And I think the ZipChip comes in here as a very useful tool because now we see that we can measure, like, for example, 96 samples a day, and we have the data available the next morning.
Excellent. Excellent. Next question I have, I noticed you showed data described as with additive or without additive. Can you elaborate on when and why you chose to use the additive or not?
Yeah, I can elaborate on that. So I typically use without additive now in our current workflows just because it's easier. Although the with additive, of course, is also not very hard to do. You just add the additive and that's it. I would typically recommend it, like Erin said, based on the type of MS system that you have. So if you have a time of flight, maybe use without additive, but with those biopharma option or bit traps, you can just do with additive.
Excellent, excellent. Thank you both so much. Those are some really great responses. Thank you once again. And to our audience, I'd like to let you know that this webinar will be available to view on demand in the next 24 to 48 hours. So please keep an eye on your email. You should get a link that you can watch the entire webinar, so the presentations and the Q&A session. And you can share the link with your colleagues who might not have been able to join us on the webinar today. So with that, I'm going to bring things to a close. Many thanks to you, Erin and Ruben, for such great presentations and taking time to answer some questions. I'd also like to thank our sponsor for this event, so thank you, 908 Devices. And I'd also like to shout out the Gen Multimedia Group for hosting this event. So thank you, David Mosley and Reynolds Gutierrez. And most of all, thank you so much to you, the audience, for being here. From all of us here at Gen, thank you and have a wonderful day. Goodbye for now.